now we come to the final talk of this session. And yet another very successful former PhD student of Andy Knoll, and that's Professor Nicholas Butterfield. So he's a professor at Cambridge University, and he has been instrumental in developing methods in obtaining microfossils from really tough rocks, where it's almost would have been impossible to get some fossils out. Also some very interesting new forms. And also he's known, as I heard, for presenting bold theories about evolution of life, and that will be demonstrated here. Please.
Thanks very much. Thank you. I want to start off really by joining the list of other people who are congratulating Andy Knoll. Very many congratulations. We're very proud of you. Keep up the good work. I'd like to say that I was there early on. I was in the first tranche of Andy's graduate students. I was there, and I remember it was really exciting times. I turned up just a naive kid, thinking I was going to study the evolution of life by looking at some fossils. And it turned out to be much, much different than that. In order to do that, you had to study the evolution of the atmosphere and the evolution of the oceans and the evolution of the whole planet. All of this stuff had to go into the equation if you're going to make any serious sense of the business. Everything is connected to everything else. And that's what I took away from that early experience. And now it's commonplace. We understand that things are connected to one another, but that wasn't the case 30 years ago. And it's turned into standard science. And I think that's why we're all here today. Anyways, what I want to do today is carry on with that theme and talk about pumping and swimming through time. And in particular, the geobiological implications of propulsion. I'm really interested in the capacity for organisms to move around. I think it's very important, and it hasn't been talked about enough quite yet. So let's just take a look at where we're going. Well, first of all, I'll put this slide up here. This is Andy Knoll's a research area as much as anything else. He's worked pretty much through this entire column here and made major contributions throughout. This is a really interesting diagram, and we've seen many like it. But the point is that we've got deep time on the one hand, and we've got tectonics, we've got atmosphere, we've got ocean, we've got life contributing to this system. We've got the great oxidation event. First eukaryotes turning up. There's some climate going on. There's animals appearing. All sorts of things. Everything is connected to everything else, and this is an historical account. It's our business to understand it and figure out how it works. So let's do that. Let's take a stab at doing some of this. I'm going to focus on a phenomenon of propulsion through time and ask whether it contributes anything useful. This is here's the LECA down here. This is the last common ancestor of eukaryotes and its constituent groups. We've heard a lot about what constitute eukaryotes, and they've got a cytoskeleton, they've got mitochondria, they've got a nucleus and so on, endomembrane systems. There's another thing that's really, really important here, is they also have, the last common ancestor of eukaryotes had a flagellum. And this is a really interesting little bit. And it does remarkable things. This is remarkable photography from Kirsty Wan. I think this has a whole lot to say about what the subsequent evolution of eukaryotes can get up to. So what those flagella are, they are propellers on the one hand and impellers on the other. They really allow things to move around in ways that they wouldn't have otherwise. This is from Kirsty Wan again here. It's a green alga swimming around with 8 of these flagella. And then here is Thomas Kjorboy photographing, videoing dinoflagellate. Just You have to appreciate that this is a really interesting phenomenon. And you know what? Prokaryotes can't do it. They can kind of do it, but they don't do it like this. And then, so there's single flagella, and then you can collectivize that effort. So this is just fantastic. Again, this is from Kirsty Wan. This is a hugely slowed down video of Stentor. And it's multiple flagella, these 9+2 flagella, doing this amazing thing. And that one of the amazing things that they can do is generate really enormous vortices relative to their size, right? And they're moving water around, they're propelling water in ways that, again, prokaryotes simply can't do. There's something interesting about the ability of a single-celled organism to generate currents like this, okay? And I think it's going to contribute to how we understand how the world goes round. That Stentor thing was a benthic thing and was pumping water and moving water around. But if you want to be a mobile thing, you can use multiple cilia to swim around like this, okay? This is a really interesting phenomenon. It allows you to do things that you couldn't otherwise do. These are pretty large as well, these ciliates here. So this is a figure from Kirsty Wan, recent paper, just looking into this business of well, we're talking about cell size on the one hand and the motility of these things on the other, okay? When you're talking about cell size and velocity, you have to think a little bit about hydrodynamics, okay? So there's going to be Reynolds numbers on the one hand and Peclet numbers on the other, okay? This is a depiction of Peclet numbers as much as anything else. And what we're talking about is length dimensions on the one hand, velocity, and Reynolds numbers with the kinetic viscosity of the fluid you're moving in, and then Peclet numbers with respect to diffusion, okay? So we're talking about biological propulsion as much as anything else. Here we've got cell size on the x-axis and the swimming speed up here. What we're looking at is that, to a first approximation, all prokaryotes fall into this realm down here, and the ciliates and the flagellates are in this realm up here, okay? Peclet numbers, what you're looking at here is Peclet numbers. With a very low Peclet number, this is minus 100, all exchange takes place fundamentally through diffusion. Okay, that's the diffusion dynamics that dominates. If you start getting flow, if you start getting fluid velocity, then you're talking about advection. That's primarily responsible for the transmission of materials one way or the other. Gas exchange, for example. Food delivery, for example. So there's Peclet number of 100, Peclet number of minus 100, and a Peclet number of 1. So we can look at this diagram here. Above this line, we've got Peclet numbers of greater than 1, so looking like this, right? And below this line, everything is fundamentally moving by diffusion. All the prokaryotes fundamentally are in diffusion world, okay? And the flagellates and ciliates, they're in this Peclet number of 1-ish or so, but they're limited by their cell size, so they're not getting up into that realm there, okay? This is Reynolds numbers now, and Reynolds numbers, it can be in an inertial world or a viscous world. An inertial world is if you throw something and let it go, it keeps on going. In a viscous world, it doesn't, right? And very small length scales. You live in a viscous world, okay? So these guys are in this, these flagellates and ciliates are in this interesting intermediate world. So let's move up then. Let's move into multicellularity. Multicellularity is a really interesting phenomenon in this propulsion business, right? We can have multiple cilia on a single beast, but that's kind of limited. But here's Volvox generating some really substantial vortices of hydrodynamic movement. And here it is bobbling around. And it's a fascinating thing, right? There must be some enormous, well, there is an enormous advantage to this because we're getting up into higher Reynolds numbers and we're getting up into higher Peclet numbers, which is a really good thing for gas exchange, for example. So that's a bit of a preamble. What I want to move on to is the capacity for flagellated multicellular eukaryotes to really get serious about pumping water around. And I'm going to focus on sponges. Sponges don't have muscle. Sponges are fundamentally just pumping machines, right? And then they have a really wonderful anatomy. It's a set of very fine channels with little compartments here called choanocyte chambers. They're loaded with individual choanocyte cells, and they all beat their flagella in unison. And there's this fine manifold of channels that goes through it. There's hundreds of thousands of these choanocyte cells per cubic millimeter in a sponge, okay? The thing is, on the outside of these sponges, which have large length scales, we end up with high Peclet numbers, high delivery rates. But on the inside, because it's this manifold system and everything is so finely divided, everything is by diffusion. Okay? It's a really neat technique of exploiting high flow on the outside and low flow on the inside. And it has huge implications for how these things work. This is just a wonderful video. I always show it. I can't get over how wonderful it is. This is a sponge in the background, and this is some fluorescein dye. And sponges, they sit there and be kind of sponges, and you don't really think very much of them. But there goes the fluorescein dye. And there comes the fluorescein dye, barrel-assing out of those things. It's amazing, right? That's just a bunch of flagella banging around in unison doing their thing, right? It's amazing what those things can do just by virtue of collectivizing the power of the individual flagella and making things happen, making water move. It's coming out of here at up to 20, 30 centimeters a second, right? It's amazing. It's not only amazing for moving water around, what happens to that water is also equally important. This is an extreme end member, of course, but still, let's look at it. 1 square meter of sponge reef can process in a 24-hour period 250 cubic meters of water. A column of water 250 meters above it in one 24-hour period. It's amazing. And when it does so, it removes 90% of the picoplankton. It's a serious water scrubbing apparatus, right? And it removes a whole bunch of the DOC. And here's a quote from de Groot from a few years ago. These sponges remove the same amount of DOC in 30 minutes as free-living bacteria in 30 days. I don't know if that's true, but I read it in a book, and so there you go. So, when do sponges first turn up on the scene? It feels to me like they've got to be part of this carbon cycling kind of thing, right? Well, I don't know if this is true or not. I'm a famous skeptic when it comes to this stuff, but Liz Turner, just a few months ago, published an account of some possible keratosin sponges from the Tonian, from the early Neoproterozoic. I don't know. We have to take it seriously. It's a funny fabric, to be sure. This is a modern counterpart here. I would like it to be true, kind of. One of the reasons I'd like it to be true is because and Andy is exactly right. You know, everything is connected to everything else. I'm a big fan of this paper from 2011 from Eli Zipperman and a bunch of other Harvard regulars. It's the perfect title. A biologically induced initiation of Neoproterozoic snowball earth events. Look at, there's got this cryogenian world coming along. What in the world is inducing it? Somehow you have to make it happen. And well, one of the ways of making it happen is to change the nature of the carbon cycle. And one of the ways of doing that is to change the nature of the things involved with it. And we've already heard a bit about those V-shaped microfossils, about those scale microfossils. There seems to be something happening in the Proterozoic world around about just before the Cryogenian, right? Now, Andy, in this paper, and I suspect it was Andy's contribution, suggested, well, look at, there's these new kinds of protists coming in. And there might have something to do with carnivorous eukaryotes preying on other eukaryotes and that feedback kind of system. I'm not overwhelmed with that explanation. what about, because I don't think that doesn't feel like it has a big enough kick to me, but what about if you put sponges in the scene at about that time? I think that would have a kick. And that might do it. I don't know if this is right. It's probably not right. It's the right way of thinking about things, if you ask me. Anyhow, in some sense, none of that matters, I don't think, in the grand scheme of things, because all that's on the ocean floor. Sponges are fundamentally benthic. I think we need to get up in the water column and really start to think about what's happening, what makes the water column change from, say, one state to another. What you're looking at here is, this is an experimental lake in northern Canada, and there's a barrier across here. And you know, over on this side, the water is beautifully clear and is dominated by diatoms and other eukaryotes. And over on this side, it's perfectly turbid and dominated by cyanobacteria. And you know what? The conditions on both sides are exactly the same in some fundamental way, okay? So here are the conditions. Here we're talking about alternate stable states, okay? The same amount of nutrients are going on both sides, the same kind of oxygen in the atmosphere, everything's the same, but they're occupying differing alternate stable states, okay? This got tipped into this cyanobacterial world by dumping a bunch of nutrients in, but then you pull them off and it just sticks there, okay? There's a hysteresis in the system, right? And that fascinates me because this smells like protozoic world and this doesn't. And how do you get over the hump is the question. And when you get over the hump, one of the ways of getting over the hump, of getting out of that turbid world, you know, is to hoover all that turbidity out of there, okay? And how do you do that? Well, I'll tell you what, suspension-feeding animals are really, really good at hoovering. We already saw it with the sponge. Here's a nice little example from Denmark. There's a Rinkelbæk fjord. It's 50 kilometers long. It's not very many meters deep. It's it was famously a bit of a cesspit. And pea soup, cyanobacteria-dominated cesspit, right? So look, we have to do something about this. Let's open up this sluice here, and they did so in 1996, 97. Let in a bit of seawater, enough seawater to allow these soft-shelled clams to get going. And look what happens. There's the sluice opening up, and here's the chlorophyll just crashing right out. And this has gone from cyanobacteria-dominated to diatom dominated, and this is filled up with all these suspension feeders, okay? It's a big deal. It's a top-down deal. It's fundamentally shifted the whole ecosystem from one state to the other, right? And there's lots and lots of these examples that go on all day, but the Great Lakes have experienced something similar with the invasion of zebra mussels and quagga mussels and so on. It's just switched over. These are super oligotrophic lakes now, and it's really bizarre. It's not really bizarre. It's just a bunch of really powerful suspension-feeding, pumping kind of benthic creatures have gotten into the system. I want to move on to the biological pump. And this appears in every textbook, of course. The biological pump is the business of sequestering atmospheric CO2 by phytoplankton of one sort or another. And most of it recycles up in the upper water column, but some of it trickles down and ends up in the long-time geological archive. It pumps CO2 out of the atmosphere and into the sediment, okay? I want to introduce a new kind of tissue. We've got those in sponges. It's a fascinating collectivized activity with all of those choanocytes. But this is a much more clever trick. These are individual cells, are muscle fibers, and you can. It's just upping the game entirely to switch to this new technology, which is collectivized muscle tissues. And this is some really wonderful imagery from Thomas Kjorboy again. Muscles allow you to do a whole bunch of things that you can't do with flagella, right? And we're talking about these swimmers over here, the zooplankton, the pelagic predators, and the deep consumers, and so on. But this is something that you just cannot even think about doing at a cilia kind of level, right? And it changes the rules of the interchange between the biosphere and the hydrosphere as much as anything else. And here's the thing. These guys here are getting beyond this realm of Reynolds numbers. So this is all viscous world back here, inertial world up here. These guys are breaking out of viscous world and going into inertial world. And it's a big, big deal, right? And it has real serious implications for the carbon cycle and so on. So we can go back, well, so there's these larger things over here, and we can just take a look at them. These are muscle-powered devices now, and they have a really big impact on things. One, for starters, they're up in the water. Okay, you can't properly swim as a large thing until you've got something like muscle, right? When you're up there doing your muscle-powered stuff, you have a capacity to process enormous amounts of water. Salps are about as close as you can conceive of as far as a pelagic sponge. They have capacity for really capturing fine particles in the water column. And you know what they do? They turn it into turds, and they package it up and they send it to the bottom. And that's size really matters in the carbon cycle, right? And salps are one of the big players in the modern carbon cycle. And you know, the other one is krill, of course, are big players in the carbon cycle. They're packaging up small-sized things and turning it into bodies, turning it into turds, and removing biological oxygen demand, amongst other things, from the water column and sending it down to the bottom. Fish are a big deal, and they're adding, not only they have muscular mobility, they're adding absolutely enormous amounts of biomass to the whole system. This is a really very worthy paper. It's just come out in the past year, Hatton et al. Here's what. This lower chart is unsurprising. Namely, the vast majority of individual organisms are small, right? These are log charts, and most organisms, of course, are tiny, and there's not very many large things. This is the more interesting one up here, which is that when you tally it all up, most of the biomass in the oceans today is not the small stuff at all. Most of the biomass is fish. That doesn't mean that fish are doing massive amounts of metabolism, because there's this 3/4 scaling law. It's not 3/4 and it's not a law, but you get the idea. Most of the biomass in the oceans is fish and zooplankton and whales. And they don't have high metabolic rates, but you know what? They're doing something really weird and crazy with that biomass that wouldn't have happened otherwise. Here's one of the things that's weird and crazy about what those guys are doing. All of that biomass is ordered in a really fascinating way. It's ordered in a tiered way. This is this is a food web cartoon from the North Atlantic. There's primary productivity down here. And these are just layers of consumers, okay? This is the first layer of consumers, and then the second layer, and so on and so on. It's all of that biomass of zooplankton, of the consumers fundamentally, is ordered in a really interesting way, which is to say that body size gets incrementally larger as you go up that food structure. Biomass, that's where all the biomass comes from, is those big things. And there's a bunch of marine diversity comes there as well. That's what we're starting off with, is the phytoplankton, and we turn it into zooplankton, and we turn it into fish. Okay, and this is where all the biomass is up here in a really serious way. Most of what you're looking at here is fish, and most of them are predators. Most of them are visual predators. This is a scheme, by and large, a big fish eat little fish, eat smaller fish. Eat zooplankton, eat phytoplankton. Bruegel had it more or less right, okay? That's where most of the marine biomass is sitting. Remember, it wasn't there in the Proterozoic. That large size, they get ever larger as they go up here. That large size gives you, the larger you get, the more you can move around. And the more you can move around, the more you move that biomass and all the associated nutrients around. The more you move around laterally, the more you're connecting up local ecosystems on a regional basis, on a global basis, and so on and so on. And so here's phytoplankton moving on a scale of meters versus zooplankton tens of meters versus these, as you go up the trophic structure, they're moving more and more. They're getting larger and larger. This motility thing is really fascinating. This is a fascinating phenomenon. I'm in awe of it. It's diurnal vertical migration. Who's to say how much? Let's make up a number. 50%. It's not well known, but some large fraction of the biomass in the oceans today engages in diurnal vertical migration, which is to say that during the day it dives down to depth and stays around at depth, and then at night comes up and feeds and does it, just like clockwork, right? And we know now what it's all about. It's about visual predation. In the absence of visual predation, it doesn't happen. It's to get down into the dark and avoid the visual predators. And it's, everything is diving down and following. It's just an absolutely fascinating phenomenon. And everyone does it. Here's krill doing it. Here's eels doing it. Here's big-eye tunas doing it. Everybody does it. You go down to avoid the predators, and the predators follow you, and so on and so on. Keep in mind that we're talking about most of the biomass in the ocean. This isn't some afterthought that's sort of stuck on this. This is most of the biomass in the oceans. It's like trees on land. They may not be metabolizing huge amounts, but they're moving stuff around like nobody's business. And small things don't move around at all, right? We have to figure that into the equation. Oh, here's some elephant seals following all of that. And the whales, of course. Whales are hugely important ecosystem engineers. They're going down and tracking and feeding on those deep sea things. And it's feeding back in all sorts of metaphorical ways onto how the system works. I'm going to wrap up in a minute. I haven't got a foggiest idea where I am time-wise. Oxygen minimum zones are really interesting. Oh, do I? Oh, I see. It's 9 minutes left. Oh, excellent. Oh, okay. Okay. Oxygen minimum zones are a really interesting phenomenon. Okay, and here they are outlined on our modern planet, and they're an interesting interplay of productivity on the one hand, upwelling, productivity on the one hand, and various other factors on the other. I'll come onto them in a moment. But there are these enormous wedge-like zones of low oxygen, going down to very low oxygen levels indeed. And it's oxygenated in the surface and it's oxygenated below. And because there's a lot of productivity, especially in the upwelling zones along the margins of continents and so on, you have a lot of biological oxygen demand and ends up in those oxygen minimum zones. So that's all well and good. Here's something. I think this made my day when I came across this paper a few years ago. Bianchi et al. 2013. Intensification of open ocean oxygen depletion by vertically migrating animals. And the punchline of this is, look at what is it that defines the tops of these oxygen minimum zones. Anywhere you go. All sorts of data here. It's not entirely clear here, but the punchline was that the top of the oxygen minimum zone is coincident with the bottom everywhere of diurnal vertical migration. Okay, which is to say, what you've got, what's happening, and I think it's fair to say, is that all of that biological oxygen demand that's being produced up in the surface waters is being picked up by those vertically migrating beasts. They're coming down and hanging around during the day in this oxygen minimum zone, where it's dark, where there's a minimum amount of oxygen. You can keep a certain range of predators out by virtue of that. They spend the day down there, and they metabolize it out, and they shit it out, and they die it out, and it consumes oxygen at this level. And then the next day, or the next night rather, they go up to the surface and they do it again, okay? And that's why the top of oxygen minimum zones coincides with the bottom diurnal vertical migration, and it works even out in the deeper ocean where there's less nutrients, and the water is clearer to a deeper depth. So DVM goes that much deeper out in these less nutrient-rich waters. I think it's really interesting. And here's the thing: I think you can. There's a nice analog to it as well. Namely, you have to take there's your biological oxygen demand, and if you want to have nicely oxygenated surface waters, then you have to clear that out of the surface waters. Otherwise, you're going to end up with really messy sorts of neighborhoods, right? If you want nice, leafy, high-end neighborhoods that are beautiful to look at, you have to have daily rubbish removal, right? That's what all of those creatures are doing. There's krill, there's salp, there's copepods. They're up there hoovering out all of that productivity from surface areas. They're putting it in a truck and they're driving it down to depth. And they're doing that every day, dumping it in that oxygen minimum zone. That's why those oxygen minimum zones are there, right? Okay. They are a byproduct of daily rubbish removal. And it wouldn't happen unless you had all those animals doing all the hoovering, right? And all the trucking. And those guys weren't around for most of Earth history. Keep that in mind. Most of the biomass in the oceans, what we think of as a normal ocean biomass, wasn't there for most of Earth history. So I want to now just go back and take a look at whether any of this makes sense in terms of Earth history. And most of the action is going to be up here. So let's zoom in on that. We just heard a little bit about Ediacaran things. They're very strange creatures, but you know what? Their Reynolds, at least on the outside, their Reynolds numbers have gone way up, right? And their Peckley numbers have gone way up because their length scales have gone way up, and they're generating turbulent currents and so on. There's been a lot of discussion about how these things work. There are good examples to show that these rangiomorphs were hollow, and I think that's very important. They had compartments on the inside that were very low Reynolds numbers and where you can do external digestion. But one of the things to appreciate, almost certainly, these things are certainly eukaryotes, and they would have been covered with cilia, probably cilia on the outside, cilia on the inside, doing all sorts of things to make these machines work as far as gas exchange and as far as feeding is concerned. You can move into the Cambrian. The Cambrian is muscle world, okay? As much as anything, all of a sudden things are starting to move around in a serious way, and they've got all sorts of apparatus. They're just bits and pieces of Cambrian arthropods. The Cambrian is famous for its arthropods, and they do visual predation, and they've got wonderful screening apparatus, and they've all sorts of tools, muscle-applied tools for making things happen, and they feed with those, and they turn it into fecal pellets. And so here are some Cambrian fecal strings, and they're changing the nature of the biological pump. In the Cambrian world, we can go up a step. We're building that tiered trophic structure. There are bigger kinds of muscle-powered devices in the Cambrian. Anomalocaris it's a visual predator. Odoria is obviously a visual kind of thing. It's not clear that this thing didn't have eyes, but it sure as hell got a propulsive tail, right? These things constituted substantial amounts of biomass. And they were doing something substantial with respect to other organisms in the ocean, and they were moving it around. They had a capacity to move it around, and movement, to my mind, is the big player here. Here's the Cambrian, there's the Ordovician. There's a great Ordovician biodiversification event. There's also, it's a propulsion radiation, right? I mean, there's a whole bunch of seriously new kinds of ways of moving around going on as you move from the Cambrian into the Ordovician. Maybe not fundamentally new ways, but they're sure tapping into the new technology. There's graptolites, conodonts, and cephalopods, graptolites, conodonts, and cephalopods, and even in the benthos, because there's all sorts of new suspension feeding going on. And this is a big, it's only so many times I can repeat it. It's a big thing. In the Devonian, there's a real revolution in fishiness in the Devonian. Okay, you start getting really, really big fish. Okay, Devonian is the age of fishes, and this is from Christian Kluge and colleagues a few years ago. Through the early Paleozoic, we get enormous kind of plankton sort of signal, and the graptolites go out, and the swimmers come online. That's a big, big player. The fish, the swimmers, are the drivers behind that diurnal vertical migration. Here's cephalopods just increasing their swimming velocity, increasing their maneuverability as we go through the Devonian. Okay. There's our modern marine food web. Things are getting bigger as we go up through this system. They're getting bigger and faster and deeper diving. Predators are mostly visual predators. They're forcing stuff to move, or they're moving around themselves, and they're forcing everything else to move around. As a consequence, they're changing the nature of the carbon cycle, I would argue, and the nature of the expression of oxygen minimum zones. Here's big fish eating little fish. This is the modern world, and there's 500 meters of diurnal vertical migration in the Red Sea. But this thing didn't evolve overnight. This thing started off down here and built itself up incrementally, right? So this is just made up, right? Let's go back to a time when those pinnacle predators weren't around, okay? The really big, really fast ones, really deep diving ones. They came later, obviously. Let's go back to a time when they weren't around. In that case, this diurnal vertical migration is going to be that much less, right? And we could go back to the Devonian. It's going to be that much less again. And we go back to the Cambrian. There's only these little pissy-ass things swimming around, and they're not going to drive to great depth. In the Ediacaran, it's going to be nothing at all, okay? Worth speaking of, as far as we know, there's nothing much swimming around. Here's the thing, is that without visual predation, you don't have daily rubbish collection. It's not getting moved out. And so you know what's going to happen? You're going to have the top of the oxygen minimum zone is going to be up in the very, very shallow water. And you know what? It might not have anything to do with all this oxygen. These redox proxies that we're thinking about, they may be a reflection of this kind of phenomenon rather than the actual concentration of oxygen in the atmosphere. I don't know. I'm just being contrarian, right? Here's another thing that happens, is that and we've heard a little bit about it already, when you hit a mass extinction, all of those large, fast-swimming, deep-diving things that force diurnal vertical migration, they all check out in mass extinctions. And, well, this is simplistic, right? But we ignore all those fish in the sea at our peril. I think they're really, really important contributors to this system. And when you knock them all out by mass extincting them, stuff is going to change, right? Anyhow, I'm going to stop there. Everything is connected to everything else. The lithosphere and the hydrosphere and the atmosphere and the biosphere and the time and mixing and propulsion. And we could go on and on and on, right? Here's Andy Knoll in 1988 contemplating it all up on Victoria Island. And it's been great. Thanks a lot, Andy.